Hey everyone, welcome to the live stream. Today we'll be going over endocrine system and immunology. First, we'll be doing the, let me see. Yeah, first we'll be doing the hormone stuff. So we'll be doing the endocrine part of it first, and then we'll be doing the immunology later. All right, so if you have the worksheet, please pull it out and we can get started. All right, so the first question we're going to be looking at how hormones function and how they regulate cell structure. So they're telling us that hormone 1 is a protein-derived hormone, and hormone 2 is going to be a cholesterol-derived hormone. And that's important to know because the protein-derived hormones are going to act differently than cholesterol-derived hormones. So we're going to be using those information to help us answer these questions. And also, it's important to note that the cells that respond to hormone 1 increase production of substance X. So, hormone 1 is something that's going to increase something, whereas hormone 2 is something that's going to decrease the production of substance. So, in medical applications, the type of hormone dictates the mode of administration. For example, oral versus injection. So, the first question is asking us, how would you need to administer hormone 1 to an organism deficient in this hormone? So it's saying that hormone 1 is a protein-derived hormone, right? So do you guys have any guesses on how protein-derived hormones are administered? All right, so we know that hormone 1 is protein derived, right? Which means it's going to behave like a protein. So, with that information, we can guess that it would be a bad idea for it to be ingested because if we ingest them, then our stomach is most likely going to digest it, and we don't want that happening because we need the hormone to actually have an effect. And because of that, protein hormones are going to be administered by injection. And you can remember that by thinking about insulin. So insulin is a protein-derived hormone, and you usually are like aware that people will need insulin shots. So I would use that to remember that protein-derived are by injection. All right, and then contrastingly with the oh, I just saw the comment that said injection. Yeah, that's the right answer. Sorry, I'm just seeing that right now. All right, and contrastingly for the hormone 2, which is did it say? Yeah, which is a cholesterol-derived hormone, we are also not going to inject it because, or sorry, we're also not going to take it orally. Um, so sorry, I got it confused with the first part. So the cholesterol one is generally not going to be digested. So if taken orally, they'll pass through the digestive system and like get put into the circulatory system. So it is okay for them to be taken orally. All right, next we're going to be looking at antagonistic fashion. So that's sometimes hormones act in that way. So one hormone will make something go up and another hormone will make something go down. And if it directly opposes each other, then we call that antagonistic. So they want us to illustrate that process using insulin and glucagon. So can anyone tell me what insulin does or what glucagon does in the body? So they both regulate blood sugar concentration, but does anyone know what it specifically does? Common says glucagon lowers blood glucose. It would be the opposite because insulin opens up like allows cells to uptake glucose. So insulin is the one that's going to be lowering blood glucose, whereas glucagon, what it does is it takes the bigger glycogen chains in your liver and breaks it up into smaller glucose. So glucagon is actually going to increase it. Insulin takes up glucose, but glucagon acts as storage. So yeah, no worries. So just to make it clear, insulin is going to help cells uptake glucose, and glucagon is another hormone. So I think you're confusing glucagon with glycogen. So remember that glucagon is a hormone, and then glycogen is the one that acts as a storage. Yeah, no worries. I usually I used to get that confused a lot too, and so glucagon has ON at the end of it. So I usually think like, oh, it's like on the blood and like hormones act on the blood. So I don't know if that helps, but you can like try coming up with mnemonics as well. But yeah, going back to this question, insulin is going to lower blood glucose level, and glucagon is going to increase. And another thing to remember is that because they're antagonistic, you can never have high insulin and high glucagon because then, like, what's the point, right? Like, one is decreasing, the other's increasing. So it acts like one at a time. So if your body needs to lower blood glucose level, then there's high insulin. And then if it needs to increase blood glucose level, then there's high glucagon level. So it's like one or the other type of deal. All right. Does anyone have any questions about that or antagonistic functions? All right, let's look at the next part, question 3. All right, so assume you are trying to characterize one of the hormones that are involved in the neuroendocrine regulation of milk production. You take extracts of the blood and mammary tissues of normal lactating animals and assay these extracts by injecting them into animals that have had their hypothalamus glands surgically removed. You are looking for a restoration of milk production. Indicate whether each type of finding is consistent with the hormone being a steroid, a peptide, or either type. Okay, so remember what we learned previously about peptide hormone and cholesterol derived hormones and how they act differently. So we're going to use that to help answer these questions. So if the hormone is found in the blood, um, which one do you think it is? Do you think it's a peptide hormone or do you think it's a cholesterol hormone? Or both? All right, so because we're looking at the endocrine system, everything like travels through the blood. So because it's in the blood, that doesn't really specify anything. So it can technically be either type because both peptide hormones and cholesterol hormones travel through the blood. And then what about the next one? It is found in the cytoplasm of the mammary tissue cells. Yep, you're right. It would be steroid. And that is because only steroid hormones can cross the lipid bilayer. So if it's in the cytoplasm, then only a steroid hormone can cross that to have the receptors. All right, and then it is associated with receptors. Similar to the first part, it is either type because both protein hormones and cholesterol-derived hormones do use receptors, but it's more of a matter of where the receptors are, whether they're on the cell membrane or they're in the cytoplasm. All right, and then the next one is it is found associated with protein complexes that contain G proteins. This one is going to be only peptide because they're the only ones that are going to associate with G protein complexes. And you can remember like protein for protein. I see a comment asking about the sign-in form. I'll be sending that at the end of the meeting, so you guys can sign that once we're done with the worksheet. All right, the next one it is found in the nuclear extracts of the cell, and like we talked about before, only cholesterol derived and steroid hormones are able to cross the cell membrane. So if the receptors are, or if it's found in nuclear extracts, then we know it's only going to be steroid hormones. All right, for the next three questions, it's going to be true or false. So let's see what they are. So again, we're looking at glucagon and insulin, which are hormones that act homeostatically to maintain blood glucose levels. And let me know if you guys think these are true or false. So question 4. Their production and release is stimulated by tropic hormones from the anterior pituitary. All right, do you think this is true or false? I'm seeing a false in the chat. Yeah, great job. This one is false because the pancreas is the one that's going to monitor levels of glucose in the blood. So you can remember beta cells are the insulin one, and I believe it's the alpha cells. Yeah, alpha cells for the glucagon. So false. All right, how about this? Oh, we already looked at this. This is true. They are antagonistic hormones. And the last one, they act by stimulating storage or release of glucose from cells. And this one is true. So we know insulin stimulates storage of glucose, and then glucagon is going to release glucose from the cells. Okay, any questions so far? we're going to be moving on to the immune system after this. All right, looks like I haven't gotten any questions, so let's look at the immune system. So the first question is, what are pathogens? Why do we need to prevent them? And what effect they can have on our body. So would anyone like to tell me the definition of a pathogen? All right, let's see. So pathogens are going to be is a general term too. So pathogen is just a really broad term for any organisms that can cause disease. So we can say pathogens are disease-causing organisms. And they include like disease-causing bacteria, virus, fungi, protists, so it just encompasses pretty much everything. And another thing is like these components, like the other question asked, like why do we need to prevent them from colonizing our body? Because some of them can make us very sick, and they can also compete with our own cells and potentially destroy them too, because they need the same resources that our cells have. So they can be very harmful, and they can have a bunch of different effects based on like what type of pathogen we're talking about. So for example, they can like paralyze some of our cells, like if they destroy a lot of cells that we need, like they can do that. They can also produce a lot of toxins, which also destroys our cells. They can just overgrow and put like too much pressure on our membranes and like cavities, which can cause um, like in the sinuses, like pressure builds and it can cause like pain or blockage or like draining of different fluids. So yeah, that's why it's important that we don't let them take over our body, and our body has a mechanism to do that, and that's going to be like our immune system. So we're going to be looking at general defense. So that's going to include the skin and the mucous membrane, and those are called physical barriers. I'm just going to write that down. Also, this is called general defense mechanism because they're not specific to any bacteria. Because you know how like antibodies are very specific to the type of disease or pathogen we're dealing with, but physical barriers are going to be activated no matter the pathogen. That's why we call them general. All right, and another type of general defense mechanism is going to be lysosomes that are secreted, and also acidic pH in our stomach too. It's very acidic. That can be considered as another defense mechanism. And another, oh, I have a question. So it says, what about platelets, neutrophils, and macrophages? So those are going to be general ones because they are not specific, but right now we're just looking at inflammatory responses, and they'll be useful in that, so I'll get to that. But you're right that it is a general type of defense. Okay, so the one we just looked at is like physical barriers, and now we're going to be looking at inflammatory responses, and this is when we're going to be dealing with those white blood cells that was mentioned in the chat. So when a localized injury occurs, there's release of prostaglandins and histamines, and those are released by white blood cells, and that causes an increase in blood flow and swelling in the area. And we also have clotting mechanisms, which is where like the platelets come in. So we have clotting mechanisms, and another reason that inflammation is actually a good thing in when we're harmed is because we want our blood vessels to vasodilate so we can have more of these factors flowing to the site of injury so they can fix them. And this is where we'll have our phagocytic white blood cells move there so they can swallow up the pathogens, and where we would want our platelets and other clotting mechanisms to go to. So that'll be our inflammatory response. Okay, any questions so far with the general immune response? All right, let's look at some specific immunity that our body has. So the two main things are going to be B cell response and T cell response. And we're going to be looking at the difference between that. But the main thing to know about the B cell response is that it's going to, well, both of these actually. So the B cell and the T cell are going to respond to specific types of pathogens, foreign pathogens, and B cell specifically responds to the antigen, and it secretes antibodies that are going to interact with the antigen and causes the cells, the pathogen cells, to clump together, and agulation. And agulation means that it's going to tag or identify the cells. So then some other like phagocyte cells can identify that it's not a cell of our own body. So B cell, I would mainly remember that it responds to specific types of foreign antigen. And most importantly, it secretes antibodies. And I would know that what that actually like does, like other than producing antibodies, the antibodies are going to interact with the antigen, causing the antigen to clump together and tags it. One of the comments says, and B cells collectively are referred to as the humoral response rate. Yes, that is correct. And the reason why they call it humoral response is because it's happening through fluid. So it's occurring through our circulatory system. So that's why they call it humoral response. And T cell response is going to be called cell mediated response. So I can write that here too. All right, so for the T cell, it's going to be a little different. So when the body becomes infected with pathogen, foreign protein from the pathogen interact with MHC molecules, and the MHC antigen complex is going to migrate to the surface of the cell membrane. So just to like visualize it, so say this is a cell, then it presents something like some random proteins, and that's what we mean by it's going to migrate to the cell membrane surface. And what's going to happen is that the T cells are going to individual T cells are going to have like complementary antigen receptors, and they're going to bind to it, and then they're going to display the MHC. And then the cytos and then this is where the function of the T cells differ. So we have the cytotoxic T cell where it's going to bind to the protein, and then there's going to be the helper T cell, which is going to display MHC2 on it. So important things to know here is cytotoxic T is going to bind to class 1 MHC, and then helper T binds to antigens displayed on class 2. So I would know which one interacts with class 1 and which one interacts with class 2. And then another thing for the cytotoxic is that it can directly kill infected cells. Another name for it is killer T cell. So you can associate that with directly killing the infected cells, whereas the helper T cell, its job is to help our immune response. So it helps with dividing and producing other helper T cells. It activates memory T cell, and then memory T cells are going to remember it later. So if you have the same infection again, then your cells will already have them in store. So think of cytotoxic T cell as killing, like actually physically doing the killing of the infected cells, whereas helper T cell is more about recruiting other members to play a role. So memory T cell, helper T, like more helper T cells. All right, any question in the difference between B cell response and T cell response so far? All right, let's move on to the next question. So this is asking if 10 to the power 5 genes are available in the human genome to produce proteins, how can we produce more than 10 to the 12 power different types of B cell receptors on B cell? So it's basically asking, earlier we talked about how B cells need a specific antibody for antigen, right? So they're basically asking, how is there so much variety? How can it produce like so many different B cell receptors? And the answer is random recombination. And this is in the production of B cells. So when you're producing B cells, there's just a bunch of random recombinations that are occurring. And those recombinations is what allows you to have different kinds of B receptors. And it essentially goes back to how, like, the antibody has, it's also important to know that antibody has, like, like different regions, and there's a variable region that varies, but then there's also a region that stays similar. So the region that stays similar is going to stay the same no matter what type of antibody it is, but only the variable region is going to be made up of three parts, and they're coded by different regions of the DNA. So when that part is being coded, that's where you get the random recombination. All right, so the last question is, how does HIV affect the immune system? So HIV is a little different. So it's called human immunodeficiency virus, and it targets cells that display both CD4 receptors and the chemokine receptors. And these two receptors are important because they're found on our helper T cells. And if HIV infects that and it hurts our helper T cell, then our immune response is going to be significantly lowered. Because, like we learned before, the helper T cell plays an important role in recruiting other helper T cells, memory cells, and other things that we need for a strong immune system. And this is why HIV is pretty severe, because you're not dying from the HIV itself, but you're dying from other infections that you cannot fight off. So even like a small cold becomes very severe with someone in HIV because they just don't have the immune system to help fight it off. So I'll write the CD4 part here. So HIV targets CD4 and chemokine. And they're found on our helper T cell. All right, so it was a quick worksheet today. So that's our last question. Let me know if you have any questions in the chat, like if you whether it's about endocrine or immune system. Okay, does anyone? Okay, let's see. Do T cells also undergo gene recombination, or is it only B cells? That's a great question. So it's only going to be our B cells because our B cells are the only one that are responsible for producing antibodies. The T cells don't really need that because T cells are going to be reading other cells, right? So, like the killer T cell is going to recognize anything that's not its own. So all our body is going, like every cell in our body has its like own marker. So the T cell is essentially like, okay, is this something that should be in my body, or is this something that shouldn't be in my body? So it uses more of that technique rather than actually producing the complement protein that we would need to interact with the antigen. Did that clear your question? All right, does anyone else have any other questions? Yeah, no problem. All right, I'm gonna go ahead and put the extra credit worksheet. Make sure to fill it out to get the credit that you guys all deserve. Yeah, no problem. Thank you everyone for coming today. Sorry about any confusion with the time that's been going on. I know a couple people weren't sure. But yeah, just a reminder that it's going to be on Thursdays from 12 to 1. Okay, I'm gonna end the stream now. Thank you again, and make sure to fill out the form. Bye.